Our next speaker is Anqi Huang, and the title of her talk is Manipulating Fate with Light, an optogenetics tool to understand bicoid function.
Thanks, Liz. Today I'm going to talk about fate. In our life, our behaviors change the fate of our future. Sometimes even small mistakes that we make in our behavior will leave us in deep, deep regrets. That's probably how most of the British people are feeling right now. However, the story I'm going to tell today is a bit different. That is, in a developing embryo, it is the fate that predominates. And small alterations in cell-fate decisions will change totally the behaviors of the cells. For example, the embryo development of a fruit fly takes around 24 hours to accomplish. As we can see in the top movie, each cell in the embryo is doing the right thing at the right moment, as if they are receiving constant commands. In reality, we know that what they do throughout this long period of time is pretty much predetermined in the first 2.5 hours in development, when the cell fates are laid down by a combination of different morphogen gradients. In comparison, if we take away one of the determinants, bicoid, from the embryo, as we can see in the bottom movie, that the anterior cells adopt a posterior cell fate and do what the posterior cells are supposed to do, creating a symmetric embryo. Now let's zoom in on this first 2.5 hours and see how the bicoid gradients look like there. The precise measurements from Thomas Greger's lab have shown that the nuclear bicoid concentration is first detectable at nuclear cycle 6, about 45 minutes after fertilization. After that, the amplitude of bicoid gradient increases until it reaches the maximal value around nuclear cycle 11 and 12. And hereafter, the bicoid protein starts to decay gradually. So the morphogen profile keeps on changing while the nuclear undergoes syncytial divisions. Curiously, these two dynamic processes match each other well enough to give rise to precise and faithful cell-fate decisions. Therefore, we ask the question of how do nuclei in a developing embryo interpret and integrate the concentration encoded information from a dynamic morphogen? And is morphogen in different time window equally important for cell-fate commitments? In order to answer these two questions, we sought for a way to temporally and reversibly control the on and off of bicoid-dependent transcription. It turned out that one of the constructs we made works exactly the way we intended it to. By fusing a optogenetic cassette, Cry2, at the N-terminal of bicoid protein, we found that when kept in the dark, the Cry2 bicoid acts as a white-type protein. It gives rise to perfect embryonic patterning, as I'll show later, and the transgenic animal hatches as a healthy larva. However, if we illuminate the embryo with blue light, the Cry2 bicoid protein undergoes conformational change. It still binds to its native bicoid binding sites, but it prohibits the transcription of the target genes, potentially due to conformational hindrance. The illuminated embryo, as shown here, it fails to develop a head and thorax parts resembling a bicoid knockout phenotype. Here I show one of the experiments we carried out to visualize the effect of Cry2 bicoid on transcription. We utilized the MS2 technique to monitor the RNA production of one of the bicoid target gene, hunchback. On the top is a control embryo. We can see the green dots representing the newly transcribed hunchback RNA. We can see most of the dots persists throughout the nuclear interface. However, if we express Cry2 bicoid on top of a white-type background and illuminated embryo with 488 nanometer laser, we can see that the dots become much more sparse and transient. Such interruption of the persistence of transcription abolish the production of target genes. Of course, we do not observe such difference in the posterior domain, where hunchback transcription is bicoid independent. And the inhibitory effect of Cry2 bicoid is more remarkable in the bicoid null background, as shown here in the difference of hunchback protein level. So the specificity and reversibility of this construct allows us to deprive the embryo of bicoid-dependent transcription in any sharp time window as we want. Here I use the example of Peru gene expression as an example to demonstrate how inhibiting bicoid transcription affect the embryo patterning in the. This is an embryo by the end of blastoderm stage. An embryo having developed in the dark showed an even skipped pattern like this. One of the most striking phenotype we have obtained is when we illuminated embryo throughout the nucleus cycle 13 and 14. From now on, the blue bar will represent the time window for illumination. Such embryos are missing the anterior two stripes of even skipped and showed no other signs of segmentation. In comparison, if we illuminate only 20 minutes before gastrulation, a time window often regarded as irrelevant for maternal inputs to set up patterns, we see 7 stripes. However, we can still appreciate that the shape and expression level in the first 2 stripes deviate from the control. Further, the intermediate illumination time gives rise to an intermediate phenotype. This tells us that this 1 hour time window is essential for the constant accumulation of bicoid target gene production in order to reach a proper expression level by the end of blastoderm stage. Then we try to illuminate only the first part of this critical time window. This time we see EVE expression in the anterior region, but it fails to resolve into stripes, which means that this time from nucleus cycle 13 to early nucleus cycle 14 is essential for the production of repressive factors that's essential for the resolution of segments. What about even earlier nucleus cycles? People have observed bicoid-dependent transcription as early as nucleus cycle 7, but in very small amounts. In order to determine the role of early transcripts, we try to illuminate the embryo during nucleus cycle 10 to 12. At first glance, the overall patterning doesn't seem to be affected. However, if we compare this embryo to an embryo either having developed in the dark or illuminated even earlier, we find that there's an anterior shift of the patterns. This resembles an embryo of a reduced bicoid dosage. This is very interesting to me because it seems to be telling that the bicoid presence in the earlier time would increase the nucleus competency to interpret bicoid gradient in the later time. Therefore, we call bicoid in this time window as serving a pre-patterning function. So we should bear in mind that by the end of blastoderm stage, bicoid has finished its mission in setting up cell fates. Now let's look at how bicoid fine-tunes cell fate decisions along this time axis. A perfectly patterned embryo will look like this 4 hours later. The organization of the body structure goes all the way from clypeolabrum to the abdomen segments. To my surprise, illumination for merely 10 minutes before gastrulation matters to embryo development, as we can see in this image that the embryo is missing the most anterior 2 segments, that is the labrum lobe and the invagination of the stormodial primordium. And each time we prolong the illumination, we find more missing segments from the anterior to the posterior. And in most severe case, all the segments prior to the third thorax are missing. To corroborate these results, I looked at the Hox gene expression pattern and the cuticle patterns with the same time scale of illumination as in the previous slide. We can see that in the shorter illumination times, below 30 minutes, that the more posterior Hox gene are not affected, including antenna pedia, ultrabithorax, and abdomen B. However, the more anterior Hox gene, sex hormone reduced, is experiencing an anterior shift. Meanwhile, the mouse parts of the embryo showed deteriorating defects with prolonged illumination. When the illumination proceeds for 50 minutes, first of all, we can see that the sex chrome-reduced expression fades away, coinciding with the disappearance of labial segments that it marks. And we can also appreciate here that the anterior cells start to adopt a posterior cell fate, as pointed out here, the ectopic expression of posterior Hox gene in the anterior region. This is also reflects in the cuticle patterns. Taken together, this seems to be telling us that the more anterior in space the structure is, the later in time the cell fate commitment is carried out. I will come back to this point later. Here is the last experiment I will show today. If you recall the results I showed on early patterning, we know that the bicoid doesn't play a major role before nucleus cycle 10. Indeed, the embryos illuminated for the first hour after fertilization does not affect embryogenesis. However, if we prolong the illumination into the pre-patterning time zone, again, we observed a reduction of the structures from anterior to the posterior. Okay, let me summarize the above information into one diagram. If we consider the cell fate decision a continuous process, the colored bar here represents the time window required for bicoid presence in order to arrive at a correct cell fate decision. It seems that the most anterior structure is also the most demanding on bicoid presence because any small interruption of bicoid-dependent transcription would hamper its proper formation. In comparison, the more posterior region arrive at a correct decision in much shorter period of time. In other words, cells experiencing a higher bicoid concentration also require bicoid for a longer period of time. So here we observed a spatial temporal coordination of morphogen gradients impact on cell fate decision. Is such spatial temporal coordination an intrinsic mechanism, intrinsic property to morphogen gradients and can be universally applied to other morphogen gradient decoding mechanism? And is such spatially sequential cell fate determination a robust mechanism in order to set up sharp cell fate boundaries? These are the questions need our further investigation. We can discuss about this later. I first want to thank the organizers to give me this chance to present this project for the very first time. I'd like to thank my supervisor, Tim Saunders, and the lab, especially Christopher and Xiaobo, for participating in this project, and also the following persons for sharing reagents, and this project cannot be done with the funding agencies here. Thank you very much for attention. I would like to take any questions.
I guess I was wondering if so the head sections are like the most sensitive to the blocking bicoid activity, but I was wondering if you think it's actually, if it could be the role of like, so basically so it's also like those sections also reach the highest level of transcripts due to bicoid activity. So do you think it could also just be like a thresholding mechanism? Like you need bicoid activity for this really long time to actually reach this threshold of whatever the target genes are. Or like, I guess I'm wondering.
It might be relevant to this affinity, target gene affinity, that because in different region, due to different concentration of bicoid, they transcribe different target genes, and in different region that the cell fate require different dosage of the target gene. Maybe in the anterior region, they require a longer transcriptional time window in order to reach a proper expression level of some target genes to make a correct cell fate commitment.
Just a clarification. So from your presentation, you don't know how the optogenetic construct is blocking function of bicoid? Is that what you mentioned?
Yes. What we know is that it binds to native bicoid binding sites, and it seems to specifically inhibit the transcription of bicoid target genes. I think due to conformational change or clustering of Cry2, it blocks the further assembly of transcription complex. That's why they cannot transcribe target genes.
Thank you.
Thank you.